Hi, Hannah.
Hi, how are you doing?
Good.
Good. I'm gonna let some people filter in, but hopefully we can get started here soon. I'm gonna give everyone just another minute to join on, but then we'll get started.
Sounds good. While we wait, I just wanted to introduce myself briefly, Lauran. We never got to meet because I was unavailable during that meeting time originally, but I'm the co-founder of the Chapel Hill Alt Protein Project. My co-founder Deeksha is here as well, and just wanted to thank you so much for coming today.
Okay, my pleasure. Nice to meet you.
Okay, we can probably go ahead and get started. I'm sure people will filter in as the hour progresses, but hi everyone. Thank you so much for coming to the webinar tonight. I'm super excited to get started. My name is Hannah Matthews and I'm the Director of Education of the Chapel Hill Alt Protein Project. The Alt Protein Project is a student initiative supported by the Good Food Institute, which is a global nonprofit working to accelerate the development of plant-based, cultivated, and fermentation-derived alternatives to conventional meat, eggs, and dairy. At the Chapel Hill Alt Protein Project, we strive to create a vibrant and robust community for alternative protein research and innovation at the University of North Carolina, Chapel Hill.
Hey guys, my name is Jefferson. I'm the president of the Biomedical Engineering Society student chapter at NC State. We're a student-led organization aimed to connect the students to the field of biomedical engineering, both on and off campus. And tonight we'll be hearing from Dr. Lauran Madden, who is the vice president of research and development at BlueNalu. BlueNalu has the goal of becoming a global leader in cell culture seafood, and they hope to provide consumers with healthy and trusted seafood products. Lauran has a Bachelor of Science in Chemical Engineering from Cornell University and a Ph.D. in Bioengineering from the University of Washington. She has significant multidisciplinary experiences and in the design and development of novel biomaterials, cell expansion methods, and 3D tissue fabrication to generate 3D cell-based products. Thank you so much, Lauran, for joining us tonight.
Throughout the webinar, if you guys have any questions for Lauran, please feel free to type them into the chat box, and the Chapel Hill Alt Protein Project's co-founder, Sophia and Deeksha, will ask them during the Q&A, or if you're comfortable, you can unmute yourself and ask them over the speaker once the presentation is over. And if there are no questions, we can move on to the moderated Q&A. So now I would like to hand it over to Lauran to present about the science behind cell-cultured seafood.
Great. Thanks, Anna and Jefferson. So I'm going to share, I've prepared a about 15-minute kind of intro, and then I think the idea was to have mostly Q&A afterwards, but I can always go a little bit deeper into any of the slides, depending on what folks are interested in. And I wasn't, I guess from a technical engineering perspective, there are a lot of fields that come together to create, cell cultured foods. So I'll kind of touch on as you move through the development and the scale up and the manufacturing, what are some of those applications, and how do maybe even some of the classes or things that you all are doing now apply to the development work. And then we can also touch on, how can you leverage that to then potentially work on the field or to become further educated if you choose to get involved in this type of career path. But just to kick off, a couple intro slides about the company. So BlueNalu is a mission is really to be a leader in the manufacturing of these cell cultured seafood products. So currently we're focusing on finfish products, in which we take cells, directly from the fish, and then we proliferate them and manufacture them to create products that are trusted, safe, and free from different environmental contaminants such as mercury, microplastics, or PCBs. And one of our overarching strategies is really to partner with the existing food industries, not only to accelerate our own development, but also to have more influence. So as this market grows, we connect with influencers in the space to make sure that we can really reach a wide variety of markets in a global way. So with that said, I think most of you might be familiar with the Sustainable Development Goals, but our company is aligned with 10 of the different Sustainable Development Goals. Obviously, life below water being in seafood products, but also, for example, zero hunger, creating good work and economic growth opportunities, and also sustainable cities and communities. And as we go through these, I'll touch on some different sustainability topics, but we do try to keep these, the mission and then the different goals and values in mind as we do the development process, so that we're ultimately creating better products and better systems for the food production industry. One thing just to kind of differentiate the seafood from land-based animals. I definitely found it very fascinating after I started working at BlueNalu, the actual challenges to the food supply chain. So seafood is one of the things where consumption has drastically increased in the last couple decades, and it continues to increase. However, the supply, particularly in the wild caught, it is diminishing. And you can definitely see that, over the past 10 or 50 years, as different species become popular, and this really creates a lot of supply chain challenges, where the available wild supply just cannot keep up with the demand for those products. And a couple of the other challenges that seafood specifically faces, in addition to the overfishing, is of course different rising ocean temperatures, various pollutants, viruses. You'll get algae blooms or other things related to these, and that can also have a really severe impact on the availability of healthy seafood products coming from the oceans. And of course, one thing that we hope to overcome is, as the supply chain is increasingly challenged with, for example, different contaminants, including microplastics, as you can see on this nice bottom right picture of all the plastic that's down into the ocean. A lot of it is now starting to end up in various seafood products. Is that when we remove the cells from the animal and are able to culture them in these kind of controlled manufacturing environments, we control all the inputs. So we will be much more easily able to control, for example, is there mercury or plastic or other contaminants, and we can monitor for them. So versus in the ocean, the fish encounter whatever they encounter, we'll be able to very what the cells come in contact with throughout the entire life cycle of the product. But moving on to kind of more the how do they want to develop these products. So we break the process down into 3 kind of major categories. So the first is basically the cells. So the cells are the critical raw material, one might say, of the entire process. So we chose to do a non-GMO process. Of course, there are other companies that are pursuing that. So those familiar with genetic engineering, there are a lot of tools and technologies that could certainly be used enhance the process or make it faster in the future. But BlueNalu has chosen not to, partially due to regulatory concerns, but also due to a lot of consumer acceptance concerns. And really understanding who is your customer and how will you lower that bar for product adoption. And I think that as you embark on potentially designing new products, those are considerations you want to take in early on and really understand, what is the ultimate end usage, or what is the ultimate consumer profile look like, and you can design the product and the process accordingly. But the first step, all about cell lines. In the second step, this is kind of the more of the tissue engineering related step. You grow those cells at large scale in bioreactors, as pictured on the right-hand side. And then you reform those into edible tissues. And those tissues can be, either very complex or very simple. So some of the products you may have seen from other companies are more like a ground and form. So for example, a nugget or a burger. BlueNalu has chosen to recreate more of a whole muscle product. And so that is a little bit different of a manufacturing process. And rather than creating small pieces, we actually try to create larger pieces or fillet-like products. And in the last area in the product development, it's really about understanding the sensory characteristics of the product and the nutritional characteristics. At this time, we're focusing on products that are substantially equivalent to conventional seafood. So for example, if we have, a mahi-mahi taco and you compare it to one from a wild-caught mahi-mahi, we want those to be nearly indistinguishable. And this first product also is chosen, to really understand what are those consumer adoption trends and be able to have some of the lowest bars to enter into the market. So it's a swap for the same product rather than introducing a completely new product at this time. But I'll go a little bit deeper into each one. For cell line, I know that in a lot of my bioengineering projects, a lot of it was different combinations of cells and materials. And so one of the things that we do, but also other companies do, is as you're working with new species, you do have to really optimize this cell growth process to be able to create conditions, whether that's the materials they're interacting with, the media they're interacting with, or for example, the bioreactor conditions. And all of those things require a lot of not only biological knowledge, but also engineering knowledge and understanding the process and the systems. And so in the first step, we basically establish these stable cell lines. And then one thing that we like to do as a check before moving them on is not only ensure they can grow for very, very long times. So not only pass the Hayflick limit of 50 doublings, but even 100 or 200 doublings in a stable way. And then we also confirm function. So in this image, we're just showing some myofibers created from mahi-mahi muscle cells and the expression of sarcomeric alpha-actinin, which is a muscle protein. But we also do the same thing for the preadipocytes, which are the fat cells, and really ensure that we can create these stable lines that once transferred to manufacturing will basically produce the same thing over and over again without any variation. And I think one of the kind of most interesting things, and I know that there was a question from Hannah before about, kind of where is this going? Are you, what does the future of cell culture look like, or of some of these other replacements? So there's been a couple of market research studies on the market as a whole. This is kind of just showing what the anticipated market out to 2040 might look like, where basically these different cultured, whether it's meat or seafood, go from 0 right now up to potentially like 35% of the entire meat market, which is very substantial. When you think about the meat market, you have, metric tons, like hundreds of metric tons of food. So it's a very substantial increase in volume from where we are today. And you can also see another category, which is, the other category of alternate proteins, that's the different vegan meat replacements. And I think it's one thing to note that our products are not vegan. So we do have, the cells are mahi-mahi or bluefin tuna, and they're actually genetically identical, and will be, if you do a DNA test, that's what it's going to come back as, even though we do not use any other animal products in those productions. So that's just one of the key differences between those two categories. But they do see a decrease potentially in the conventional meat. And you can imagine if that trend continues, that it would substantially decrease over the next 50 or 100 years. One of the biggest engineering challenges that we have is the scale-up process. So when you think about that volume that we're going to have to create, it's not just the engineering in the bioreactor, but it's everything that goes into it, and it's also controlling the process. So from a chemical engineering standpoint, it's a very interesting challenge. One of the things that is a little bit different in standard cell culture versus the cell culture products is the raw materials used. So we have to actually have, the food grade supply chains. All the raw materials have to be verified as safe for consumption, which is different than if you demember something off the shelf. It's a different type of quality and performance that you're actually looking at. There's another couple of interesting engineering challenges, and I don't know how many of you have had your heat and mass transfer courses, but as you scale up, the mixing, agitation, and the ability to maintain uniformity across the reactor gets really challenging without having very high shear speeds. And so this is one thing that not only do we model mathematically, but then we also determine experimentally to understand the process limitations, and then to design the processing equipment to be able to be more scalable. And then the other two things that I think are both engineering, but also operations challenges. So cost reduction is, both in the raw materials as well as the process. And if you've done any process modeling, and I used to use Aspen when I was in my chemical engineering program, but we would do the modeling processes and then do the cost analyses on the factories. And so you can do a lot of interesting things in the raw materials or the process or the equipment that you choose to then actually decrease the cost of the overall facility. And for those of you who like finance and engineering, that's probably a good career path for you. And then the last one is actually safety risk. So for those of you who work in labs, one of the things that we always talk about when we talk about safety is the types of controls that you can implement. So for example, you have your personal protection equipment, you have engineering controls, you have process controls, but there's a lot of design elements that go into reducing the risk. So for example, using aseptic transfer devices when you move between a bioreactor or another vessel, or controlling the valves, but the design of the overall process and decreasing risk is definitely one of the things that is also really important when you're designing for scale up. So just a quick picture, we're currently building our facility, so those things are all on my mind about every day. But also just to note that these facilities, we're in seafood, so we're under the FDA only. and we'll have, FDA inspected GMP facility. In this facility we're building, we anticipate it being flexible enough to do multiple different species. So starting with mahi-mahi, but then bluefin tuna, red snapper, etc., as the different technologies are built upon. And also just to reiterate kind of what I said before, but that, one of the most important things when you start to scale up, and I think is not all the time thought about at the bench scale, is how does what you're doing affect the safety of an edible product? So some of the major things, we definitely had to change, the removal of antibiotics, removal of compounds such as phenol red, the removal of allergens. So for example, a lot of pharmaceutical companies use soy peptone as their media. If you do that in a cell cultured product, you now have a soy allergen. So you definitely have to be very aware of all the chemicals or raw materials going in or being involved in the process, and understand how that impacts the ultimate food safety of the product. And one of the things that we like to do with all the products is try to have them, again, substantially equivalent to the existing product. So this was a few pictures from the culinary demonstration in 2019 of the yellowtail product, where we did a kimchi, a poke, a taco, and a soup. And importantly, one thing that we always design the product formulation for is the application. So for example, if your major application is a taco, then you want to make sure you can grill it, you can fry it, and it's going to stand up to the heat, acidification, or whatever you're going to throw at it. Here are a couple of the happy event goers. But on the on the technical side, how do you do that? Probably sounds easy. One thing that we've done is collaborate with Griffith Foods on the descriptive analysis. So for those of you not familiar, you basically for each product can develop what's called a sensory lexicon. So for example, the graph showing here is, nutty, salty, sweet. And you can actually, using an expert tasting panel, quantify those. And then when the tasters test it at different times, you can actually get a quantitative assessment of how the different tastes compare from one product formulation to another. The other more, less people involved, but more instrument. There's some e-nose or e-tongue solutions. There's a lot of different texture analyzers that are available and that we use as well to compare, for example, what part of texture defines the fish firmness or the fish flakiness, and actually be able to develop technical assays to quantify that and then make sure that your formulations are achieving it. The color can be very important. You certainly wouldn't want, a strangely colored fish. Fish should be pink or white typically. And then the last thing is the nutrition. So a lot of, for example, our different formulation designs are based to achieve certain nutritional profiles. And then we use a lot of analytical testing to compare the nutritional profiles against like the benchmark product. So these are kind of all some of the endpoints that you might use when you're designing this type of product. And as you can tell, it gets, rather than looking at, vascularization or fiber size or some of these things that are classical tissue engineering, you have a very different set of endpoints when you get to food and cell cultured food engineering. Really quick slide. I think, for those of you who are looking at, whether it's a career or just learning more, I put a couple websites up here. Obviously, you're already involved with GFI. BlueNalu also has an Eat Blue educational platform that we launched that provides a lot of links to fun stories about the ocean and kind of positive news around that. And that's all I had for kind of a short intro. But yeah, feel free to follow us on social or reach out to myself on LinkedIn as well. So I guess should I stop the share, Hannah, for the question period?
Yeah, sure, that'd be good.
Yeah, so now we're moving into Q&A. That was a wonderful presentation, Lauren. Thank you. So if you have questions, please either put them in the chat or please just take yourself off mute and I will call on you. Michaela.
I would just want to ask, like, as sorry, 1 second. Sorry about that. All my housemates started talking at once. So as like an engineer working in like chemical engineering and then going into this field, do you like, what kind of opportunities do you have to be like creative and like come up with different ways to do stuff in the same way that you would in like a normal research lab when you're working on like one type of product? So I imagine like it would get kind of repetitive to develop like a certain tissue like most of the time and then say like, how do I make this tissue more like more realistic and look at all these aspects of it, but you're making like different kinds of tissue, but tasks.
Yeah, good question. Going a little bit back. So, I mean, I did undergrad and grad school. I also did a postdoc. So I spent quite a long time in academics doing more what I would call basic. I mean, some of it was translational research, but overall it was research to publish papers. And then in industry, one of the things I love is that it actually is more fast-paced and you have a lot more variety. So one thing I definitely got maybe frustrated with in terms of academic research was, as an engineer, I want to create things and I typically want them to actually be out there for other people to use, not just published in a paper. And so now doing in this field, it's really exciting because the food industry, moves much faster. So if you think about, like your basic potato chip, like how many Lay's flavors or Gatorade flavors do they come out with every year? Like it's actually a very fast product cycle. So as we think about the product portfolio of BlueNalu, it won't just be, oh, we have one bluefin product. It will be a series and a portfolio of products, both in the US and globally. And so it's actually a constant innovation atmosphere versus, some of the other environments where you might spend 2 to 10 years on a single product or process.
Do you find like the process, like I don't really understand what a bioreactor does other than give an environment to reproduce, but do you find like it's a different approach when you're making different tissues from different species? Like it's not like you can use the same process, you need to kind of start over?
Yeah, I would say so. There's similar elements. So, I mean, you're going to have a cell, you're going to have a media, you're going to have a bioreactor. Each one is going to be tailored. So when we think about food specifically, food is at a very, very different cost point than most tissue engineering products. So if you're engineering a transplantable kidney, you can probably charge someone hundreds of thousands of dollars for that. If you're now creating a fish fillet, I'm going to be able to charge, $10 to $30 a pound. So the process becomes very, very critical to the overall efficiency. So there's a lot of work that goes into the optimization of, how efficient is the feed? Can you recycle things? Are you getting good production efficiency? There's a lot of interesting perfusion technologies now being applied to even get almost like a 10x yield increase in a lot of the existing bioreactors. That's in a lot of the kind of antibody or protein product development, but as things that we can think about applying in the cell culture field as well, as we continue to increase the production scales. But I mean, it's very interesting if you like bioreactors. Obviously, with a chemical engineering background, that was part of my minor, so it was pretty exciting.
Jonah, you can go.
So I have a question about so like once you have like your source material, like your the DNA you or like cell you extracted from whatever you're gonna make, like the mahi or whatever you're using, do you have to continue to keep going back to the source material in order to create new product? Or like since you said when you test the DNA of the product you made, like the fish fillet, it has the same DNA as the original mahi, so could you use that to create further cells?
Yeah, so I guess I actually took out the slide I had on cell bank validations, but typically how it works is, so I only take from the animal the one time to initiate the lines. So say you extract a chunk of muscle, you might have, a few thousand, maybe 1 million cells. From those cells, you can develop hundreds, if not thousands of cell lines. Not all those will be what we call like winners in terms of they grow fast enough or they function in the proper way. But what you do is you go through the cell line selection process to basically select out lines that meet your performance criteria. And then those ones are the ones that we do a very large testing panel and create cell banks. So I don't know if you've done cryopreservation, but the idea of a bank is to basically create a source of unlimited supply for the cells. And those banks go through a very rigorous validation process for identity, stability, and quality. And then once they're released, then those are essentially your source for the rest of time to make the product.
Thank you.
Allie, you can go.
Does that mean that all of the products are like identical, or do you have like a bunch of different individuals they take from? Like, is it like genetically diverse, or is it all like from the same animal?
Yeah, so good question. So for example, for mahi, we have dozens of lines, and they're from different individuals. So there is, to some extent, there is naturally occurring genetic diversity, but for example, within a single product, it's going to be genetically uniform, if that makes sense. And then the other thing we do is for certain products, we have different cell types. So, when you have a muscle cell versus pre-adipocyte, they may have the same, genetic code, but their transcriptome's different. So they're actually like functionally different cells, even if they're both, even if the DNA barcoding comes back as mahi, they're actually functionally different.
That's cool. I was also wondering what the timeline of it is. Like once you get the cells, how long does it take until it reaches the market?
So cell line development for us takes about 6 months. We've developed a pretty robust platform process. So the first line, I will admit, we didn't even get cells for like 3 months from the first species we worked on. But since then, we've kind of optimized it. The process to get to market, develop the entire scalability, that takes another about a year to year and a half. So with the process development and the regulatory filings, you can be talking, anywhere from 2 to 5 years, depending on how many things you do in parallel. So it's definitely a commitment to get one of these products to market.
And what are the characteristics that you're looking for in these winner cell lines?
So I mean, the first one is just the ability to become a line. So, we might start with hundreds and only have dozens that actually make it past that Hayflick limit. The stability is very important for us because we look at certain nutritional profiles, the function. So for example, any myoblasts we choose have to be able to maintain their differentiation capacity. Similar for the fat cells, you have to actually function as the cell of interest to be selective. And then the growth rate. So we have growth rate cutoffs. And this kind of goes more into the cost modeling, where the growth rate of your cells is going to be directly proportional to the max production rate of your facility. So if you do like a facility cost modeling, there's going to be a point at which you're no longer profitable if your cells grow too slow at how the cell behavior and performance would eventually impact the overall productivity of the facility.
Interesting. Thank you. Okay, I have another question if no one else has any. Oh wait, Jonah, you're off mute. Do you want to go?
Oh, you can go first.
Okay. So most of the technical challenges you mentioned in your presentation seem to overlap completely with cultivated meat. And I was wondering if you could go more into really specific challenges that cultivated seafood is facing that cultivated meat isn't. If there were any.
I will say at the end of the day, they're very similar processes. I think the major differences are like, what is the product that you're trying to create? So like for fish, if you look at the texture of the nutritional profile, it's functionally different. So like if someone blind taste tested you and gave you a fish fillet or gave you a chicken, you're most likely going to be able to tell the difference just based on texture. So that's one of the biggest, on the product side differences. People expect, this layering, this flakiness. There's a transition. There's actually a texture transition from the raw to the cooked. So if you have like, a raw piece of sushi, tuna is probably a really good example. Like raw tuna is fundamentally texturally different from seared tuna or from like cooked canned tuna. And so to create products that can actually get the same textural profile at all the different applications is very challenging. I don't know if you've had any of the like plant-based, but typically they work in like a single application. Like a lot of them are not very versatile in terms of being able to be cooked in many ways and still perform like at a high level. And then the other thing on nutrition, omega-3s are very important in seafood, but fish actually do not synthesize omega-3s. So that's definitely one of our raw material and supply chain challenges is, how do you incorporate the specific nutritional requirements and basically develop the supply chains, but also the methodologies to incorporate the nutritional aspects, particularly for metabolites or metabolic intermediates that are not made by the cell types. And this also comes back to a slightly different challenge, but if you think about a whole animal, a lot, and I mean a lot, of your metabolic intermediates come from your liver or other organs that are actually like hormone or nutrient producing. Very few cells make anything by themselves. Like your muscle and other things they don't make albumins, they don't make cytochromes, they don't make hemoglobin. And so depending on the profiles and the attributes you want, you have to think about how can you reincorporate them back into the product in the correct way. And for us also in a non-GMO way. So it becomes very challenging, particularly when you're trying to make a very high quality product to kind of have all these process limitations, but also then try to recreate something so complex.
Thank you. Yeah, that helped a lot. And just to go off that a little bit about your feedstock. So you mentioned how like the cost optimization with feedstock is very important in your presentation. And I was wondering if you guys are, if you're allowed to say this, if you're using any sort of agricultural byproducts in your feedstock for any of these, anything in it, including these metabolites that you're talking about. I know at the we had like a global alt protein project symposium last Saturday, and the UC Davis group talked about some research going on there on this topic, and they found out that black-eyed peas have the same function as insulin, and so like that's an option for some cultivated meat processes. So yeah, I was wondering if you could maybe explain a little bit more about using these side streams for feedstock.
Yeah, I don't know about side streams. I mean, that's definitely one of the long-term goals, is to, you know, be part of that sustainability life cycle, so to speak. So to identify materials, you know, materials that we can use to help sustainability of another process, or for our side streams to then be used in someone else's process. I will say that we've transitioned from using any of the animal components to being using plant components. So that is something we have identified. I can't say too specifically, but we're definitely going to keep our eyes out for, you know, like you're saying, if there's a specific side stream of an industry that's applicable, then we would definitely seek that out.
Okay, gotcha. Jonah, you can go now.
Okay, thank you. So this is sort of going to piggyback off of that, or at least the general topic. But as you were saying that like the actual like muscle groups that you guys are growing like for the fillets don't necessarily make or like produce the hormones or whatever that you necessarily want, like the nutrients in it. Have you guys considered, or I mean, is there like consideration with using like CRISPR or like other like genome editing tools? I mean, because it's not necessarily GMOs, granted it's still very new. But so like, are there like other avenues you guys are looking at, or?
Yeah, so we don't do any genetic modification. So I will say there's very fine lines in some of the regulatory where not every regulatory agency considers CRISPR to be GMO, but things like Europe does. So depending on where you want to sell your product, you have to be very cognizant of how people define it. So like, for example, in the US, there's the GMO labeling, the biotech labeling, a lot of it's actually based on the presence of DNA. So like there are products that use derivatives of GMOs that are not labeled as such. And so you get into a lot of murky regulatory legislation. So for now, we chose not to use any of those technologies. But you could definitely imagine in the future, as things become more acceptable. And I know there's a few companies, I think it's like Super Meat and a few of the other ones, actually specifically target GMOs to enhance the cell line characteristics.
Thank you.
Michaela, you can go. And also, y'all, we can just all talk now because I'm realizing there's only 13 of us. So yeah, just talk whenever you want.
Okay. So one question I had about the muscle cells that you were talking about, muscles themselves, like, do they need to be used in order to like recreate kind of like that texture? Like, do you need to like put like strain on them and have them develop like fibers between each other? Like, can you recreate like the texture of that tissue without having like the muscles be used at any point? Like almost as if you never, like a cow were to never move and then you ate it, like would that be the right texture?
Yeah, no, I think so. It's a hard question, but yes, you can do it. It's not to say there's no tricks used, but the fundamental basis of a muscle fiber is the protein. So, and then part of what creates the texture is the alignment. So first it's the natural fiber stiffness, but then it's also the alignment. A lot of the stiffness is actually derived from the calcium cross bridge formation. So like, for example, if you have a muscle not under tension, that's a different stiffness than a muscle that is under tension. And so when they actually do this when they slaughter animals, for lack of a better thing, is controlling the state of the muscle and the chemical status to make it not super stiff. So like when you go into rigor, I don't know how much muscle biology, but essentially when you go into rigor, it creates like a super stiff muscle. So a lot of these basic things are not necessarily just on the architecture of the fibers, but they're also on the chemical structure. So when you have the same protein composition, it becomes much easier to create that stiffness. And then if you think about, for example, if you look at the plant-based ones, there's actually a lot of ones that I feel like have done a very nice job recreating texture, and that's by different extrusion technologies versus core biology. So there's a lot of kind of, both fundamental biology, but also engineering technique that you can mash together to create these new technologies that are required. It's because it's very different than like an organ. Like I don't actually need it to function. I just need it to be the same as like a single state. So hopefully that answered your question, but yes, it's possible, but it's definitely not in the same way that you would tissue engineer like an implantable muscle.
Yeah, it definitely hearing you talk about it is making me more confused about how you do this without like any genetic manipulation, do you guys use PCR? Is that I don't really know what's in the umbrella of like, what is genetic modification? Like, what are you allowed to do without being a GMO?
So, genetic modification literally means that you modify the DNA. So, like for example, if you look at what people call like footprint-free technologies, like those would not be genetic modification. Those are also very expensive and not very efficient, so wouldn't necessarily use those in a production facility. But the one really nice thing about if you do cell line selection properly and you have very functional cells, so I don't know if you worked with like C2C12 cells or any of those other types of cell lines, but they're actually very easy to differentiate. And so it's not as big of a hurdle as you might think. It actually is like the cells are naturally meant to do certain things. And so once you put them in the correct environment, they kind of just do it. I don't know how I can better explain that, but
yeah. I hope they just do that. That's great for you guys.
Could you please explain the extrusion process more in depth? And what exactly these bioinks you're using consist of? Like, are you extruding muscle fibers, so like myotubes, or like just the cell, like individual cells? And yeah, if you could just explain that more, and like how thick, I'm assuming this is like a 3D printing type deal. So like how thick are these, like the strands of cells that you're printing?
So I'm going to use an analogy because I can't really talk about like our particular process, but
Okay.
For example, if you're making pasta, you're going to start with like flour and water. You're going to make a slurry that's mostly a liquid. I don't know if anyone's made pasta here. And then in manufacturing facilities, they just extrude it. So you use, whether that's a laminating machine to make your lasagna noodles, or whether that's a dye extruder to make your spaghetti noodles. There's a lot of technologies out there in pasta, bakery, and a few other ones. I mean, honestly, some of the, candy bars are probably some of the actually coolest technologies because they're coated, encapsulated, layered. They have like liquids inside of solids. You know, it's all sorts of engineering technologies to take what normally mostly start as liquids and create them into solid layered type products.
Okay. So would you say that through this dye that you're extruding it through, would you say it's like thousands of cells within the space that you're extruding it through?
Oh yeah. Oh no. Cell density, you have like, what is it, like 500 million to 1 billion a gram.
Okay.
It's high. It's very high.
Okay.
I've got a quick question. So for like the DNA you receive, do you receive well, because I know like some companies, I think like Jackson Laboratories, like they have the facilities to house animals that they then like send specific like DNA samples to other laboratories or other companies. Do you guys do that where you receive it, or do you have the facilities to house like the mahi-mahi itself?
So no, we don't house any animals. We collaborate with typically professionals to source samples. I can't really say who, but we have a lot of collaborations with like expert aquaculture folks to source different samples of high quality tissue. One of our advisory board members is Yoni Zahar from Maryland. And there's a really great network on the university side, on the aquaculture field, because their entire life, they're trying to figure out how can we create sustainable seafood products. And so most of them actually love this technology and they love partnering because they also want to find solutions to protect the species that they love.
Very cool. Thank you.
I had a question. So you emphasized how it was really important for you guys to like know your consumer. And I imagine like, especially because this is like an emergent technology, that you expect some skepticism of the product. And I'm wondering like, how would you combat that as a company?
Yeah, no, that's a good comment. I think one of the things that we've noticed is a lot of the skepticism comes from lack of knowledge. So kind of like any new product, if you look at consumer adoption, you're going to have early adopters who, oh, hey, that's a new iPhone. Like, I want to buy it this week, even if they have like no idea why they're buying it. And then you're going to have ones who, hey, I want to see how it goes. And you're going to get kind of the mainstream. And then you're always going to have stragglers who are, you know, may never adopt the product, and you know that's okay. But I think on the educational side, we get very consistent questions around the safety. So one thing that we like to try to do is create these analogies. So for example, people, you know, I think most people have biology classes, but maybe they don't recall like what a cell is or what cells do or how they operate. And so we try to, you know, educate people that, you know, for example, yogurt or beer or other products, you know, they use cells and they grow in food facilities and they're also safe products. And you eat those every day or every other day. And then around the other safety issues, you know, one of the early, you know, pictures or images that was out there was this lab burger. And so that is very off-putting. So the imagery that's used, you'll notice in the pictures that I used, everything is plated, kind of like you would see on the Food Network, and really reinforcing the, you know, not only a safe and trusted product, but also a appealing and tasty product. And so that's one of the other elements, is really trying to enhance the positive imagery and the positive media around the foods to help with adoption. And then the third thing that we don't do too much of yet, but as we're working on the regulatory side, we will, release a lot of data in the year to come. So not only on, what is in the product, but what's not in the product, and why it, is as safe or potentially safer than existing seafood products. So from the microbial content to the metal content, there's a lot of potential benefits to the cell culture products. And so those you'll also start seeing in the next year or so, whether that's publishing papers or releasing data or regulatory documents, is just the actual factual information and data surrounding kind of the safety profiles.
Cool, thanks.
If you could give an undergraduate or a graduate student one piece of advice on how to be successful in this industry, what would that be? Or a couple pieces of advice?
Like one piece, I would say, one thing that I've always tried to do in my career is just try to find projects or positions where I really like the science. And I would just encourage people to do that. Like, if you are very interested in seafood products, like try to find a seafood company to work at. If you love, yogurt, try to go work in that field. I think it helps a lot to be very motivated, because if you're not motivated, you're not going to want to take the time in the lab to go through the hard parts. I think a lot of people underestimate the level of failure in science is 90 to 99%. So you have to be very committed and very motivated to choose it as a career. But if you like it in the end, if you find a field you like, then it's very rewarding at the end. And then the other thing that I probably learned later than I should is just not being afraid of reaching out to people. So, if you're interested in a company, even if they don't have a post, you can contact them on the contact page. You can reach out to people who work there. Like, I know I've talked to tons of people. Like Hannah reached out and was like, hey, we have this event going on. Are you interested? And not everybody's going to say yes, but some people are going to say yes. And a lot of times that'll give you the break you need to kind of get a leg up when there are internships or jobs or opportunities available.
That's good advice. Thank you. Okay, if no one else has a question, I'm going to ask another. So I know this is all proprietary, but I don't know how much of it is, because with the extrusion process, because I'm very curious about this extrusion process y'all are using. So I know, for example, Meat Tech is also using kind of an extrusion bioprinting process, and what they're doing is they're using the bioinks, bioprinting it, and then incubating so that the cells continue to grow. Are you able to maybe expand upon that? Like, is the extrusion the last process, or is there another stage after that where the cells continue to incubate?
Yeah, so there's two different ways to approach the extrusion. So the one you're talking about is actually a scaffold-driven process, where basically they're extruding, a support material for the cells to kind of live in. And there's quite a few companies that are doing that. So them, Farms, and we chose not to do that type of process for two obvious reasons. One is that when you scale, you want to have a continuous process. Having a cold step after a process is a scale out process, not a scale up process. So it fundamentally decreases your productivity. The second one is the risk factor. So there's a huge risk in cell culture if you're going to keep your products, if you're going to, do a lot of manipulations and operations and have it at a certain temperature where microbes can grow, you basically increase the risk and the food safety. And so we actually try to make it as short as possible from extrusion to packaging. So we're kind of like the opposite of a lot of people. But we think at the end of the day, we'll have, a potentially more scalable and more productive and safer process.
Okay, that is super interesting. I have not heard that before. Cool. So you guys are not using any scaffolding, correct? Okay. Huh. Do you know of any other companies using that same process to create whole muscle products?
I am not aware because I think a lot of the ones we see do use a scaffold. But I also came from like a lot of academic labs that were really into scaffold-free technologies. So, but I think it's like it's definitely the minor component. Like a lot of tissue engineering focus on scaffolds because to some extent it's easier on small scale because, hey, I have this architecture, it's already gonna look like I want it to look. So it's less of a technology hurdle. But at the end of the day, like if you look at making 100 metric tons a year, it becomes very laborious to make all those and to then store them for, I think at one point the Ala Farms was like a 7-day maturation or something crazy. It just becomes, how many pounds are you then holding in this like perfused environment in this very complicated system? It just becomes very like the engineering challenge and the monetary input becomes a little bit daunting when you're talking about like hundreds of metric tons a year.
Yeah, for sure. This is great insight. You're making me worry a little bit about the industry because, or at least more skeptical maybe, because I know Aleph, I think this was a few months ago, announced that they're hoping to be like near cost parity in like 5 years, or like they're going to be closer to cost parity than plant-based meat in 5 years. That's what they claimed a few months ago. So very interesting.
But it does depend what you think cost parity is. So meat is doing steaks and they're doing more whole steak products. So if you think about a steak product, it's, depending on what kind of steak, $7 to $15 a pound. Whereas a lot of the plant-based burgers are like, a burger's like $1 or $2 a pound. So there's still, people throw around cost parity like it's one number, but it's really a range of numbers and it's very product dependent.
Good point. Okay, well, it's 8:02 and I want to respect your time. So thank you so much for coming tonight. And I learned a lot and I'm sure everyone else did too. So I hope to stay in touch. And I just wanted to give a quick shout out. I put this in the chat to the CellAg Australia tool to everyone here. It's a really great new tool that explains exactly how over 45 STEM majors can directly contribute to the CellAg field. And there hasn't been anything created like that before. So I would definitely look into that. Cool. All right. See you guys.
Great. Thanks. Have a good one.
Thank you very much.
Thanks for coming.
Thank
Thank you.
Bye.